In this video, we're going to take a look at the 4 levels of protein structure. So we talked a little bit about this process of how we make a polypeptide through translation. So remember we said that we go from DNA to mRNA, which ultimately gets translated into a protein. But at the end of our translation, which is what this arrow right here is indicating, we don't actually have a fully functioning protein yet. What we get at the end of translation is something called a polypeptide. And the polypeptide is actually the first level of a protein structure. So we call that the primary level or the primary structure of proteins. And we can designate that by writing the letter or the number 1 and this little like degree type circle, which just means primary. So here we're talking about the primary protein structure. And this is just referring to the polypeptide. And remember that a polypeptide is just a sequence of amino acids bonded together. Now, each protein has its own polypeptide sequence. So this sequence of amino acids is going to be different depending on which protein you're making. Now, the secondary structure of proteins is where the side chains of amino acids interact. And it causes this polypeptide to either start folding into these what we call beta pleated sheets, or they form these spirals. So these side chains interact. And remember, if we look at the kind of the general structure of an amino acid, remember that we've got this central carbon that has a carboxyl group attached to it. And then on the other side of that central carbon, we have the amino group. And then we have a hydrogen coming off of that central carbon. And then we have this thing that we call the R group. And this R group are just these side chains that we're talking about here. And so of our 20 amino acids, these side chains can be very different. Some of them are hydrophilic, some are hydrophobic, some are polar, some are nonpolar. It just kind of depends on which amino acid it is. And so these these side chains, they start either attracting each other or repelling each other. And so in doing so, it causes this kind of amino acid sequence, which we typically draw out as a straight line. But it causes this kind of this polypeptide to start making these helices. And we call these alpha helices. And we can denote this with the Greek letter alpha, which looks like a little fish. It also makes these beta pleated sheets. And we can denote that with the Greek letter beta, which is, it looks like the letter B, but it's got this little tail kind of sticking off of it. All right, now the tertiary structure of a protein is where the side chains continue to kind of interact with each other, and it makes the polypeptide continue to fold into kind of a 3D shape. So for example, up here, if we had maybe like an alpha helix that turned into a beta pleated sheet and then another alpha helix, down here in the tertiary structure, we might see something like this. So we still have our alpha helices and our beta pleated sheet, but now it's kind of wrapped around itself to form this more kind of three-dimensional structure. And in some proteins, this is it. So some proteins have their highest level of structure at the tertiary level. So in some proteins, this is considered the kind of the terminal or the functional level of the protein structure. So functional proteins in some cases. Now, in other cases, we might have a quaternary structure. And the quaternary structure is where multiple polypeptides come together to form a functional protein. All right, so in the quaternary structure, you may have multiple polypeptides in their tertiary structure that are kind of like subunits of a bigger functional protein. So, for example, hemoglobin is a protein that's found in red blood cells, and it helps to form, it helps to transport oxygen around in the blood. And hemoglobin is actually made up of 4 subunits. So it's a pretty big protein because it's actually made up of 4 amino acids, 4 polypeptides, I should say, that have all reached their tertiary structure, and now they're combining together to make an overall kind of big functional protein. So we could see something like here's one polypeptide, and then maybe there's another one here, and then maybe another one here, and another one here. All right, so I apologize for my horrible drawing skills, but this is showing you multiple polypeptides that are in their three-dimensional tertiary structure, all coming together to form one functional protein. All right, so as a reminder, after translation, we have a polypeptide, and the polypeptide is the primary structure of the protein. But some polypeptides might have a function to them, but most proteins need to go through the folding of the secondary and tertiary structure levels in order to be functional. And some of them go all the way to the quaternary level in order to form a functional protein.